Hello, everyone. Thank you for joining us for this important conversation today about Duchenne Muscular Dystrophy and the program that we're developing here at Precision BioSciences. My name is Cassie Gorsuch, and I'm the Chief Scientific Officer at Precision. And I'm joined today by two really well-known folks in the field of Duchenne Muscular Dystrophy, Pat Furlong, who is the president of PPMD, and Dr. Vera Pandayan, a pediatric neurologist at Arkansas Children's Hospital. I'm going to give each of them an opportunity to introduce themselves and tell you a little bit more about who they are. And so, Pat, I'll start with you.
Thank you, Cassie, and thank you for allowing me to participate in this important session with Precision Bio. 30 years ago, my two boys were diagnosed with Duchenne Muscular Dystrophy. I'd never heard of the word before, even though my career in early medicine involved taking care of patients, but typically adults. 30% of the time that this disease happens, it happens de novo or new for families, and it was indeed new in my families. I won't talk about the genetics or the biology, but I will tell you that the diagnosis is overwhelming. Usually and typically what you hear from your physician that you're meeting with is the natural history of this disease. And 30 years ago, this physician, looking at his toes, told me the diagnosis and the natural history. He said, by the time your boys are 8 years old, they would no longer be walking. By the time they're 14, they will not be lifting their hand to their mouth, nor will they be able to hug you. By the time they're 16 or 17, they won't be here any longer. And that was 30 years ago. And it is devastating because where do you go? Who do you turn to? What happens? And in my view as a parent, and I think every parent will like, where do I go for the answer? Where is the answer, right? And then what could we do in terms of care? Is there something we could do to preserve what they have? At the time, there was nothing, very little understanding of the disease and its biology. We had a gene cloned and a protein product and not much more information. There were no standards of care. There was no protection of the heart because they felt like, or at least the medical advice was, the heart isn't involved until they're 18 or 20. We know that that's different now. And that you go home and love them. Fast forward 30 years, I started PPMD with the idea that could we, as a community, build that community broadly? Could we then really incentivize ourselves to invest in basic science to completely categorize the disease? But then could we leverage money? Could we be an advocate in Washington? We were able to do those things. So fast forward 30 years, we do have standards of care. We may have newborn screening. We have newborn screening in 3 states, maybe more. Hopefully, we'll see that decision. We have 8 therapies that are approved. We have an investment by the National Institutes of Health of about $900 million, which led to, I think, a tipping point where industry came on board. So the world has changed. It's evolving very rapidly. But today, it is the same. When the parents have this diagnosis, it is a crushing blow to the plans that they had for life. And they must figure out what the next steps are and find a trusted physician like Dr. Vera Pandayan with whom will guide them and be that guiding light throughout the process in the lifespan.
Thank you so much for sharing that perspective, Pat, and for all of the work that you and others do at PPMD. It's really amazing to hear about the progress we've made as a field, and yet there's still more to be done. So, Dr. Panda, I'd like for you to take a few minutes to introduce yourself to the audience, please.
Sure. Thank you, Cassie, for inviting me for this, and thank you, Pat, for that impactful introduction. I don't know if I could match that, but I can say my name is really long. It's Vera Pandayan, Variobi Panda. I'm a pediatric neurologist here at Arkansas Children's Hospital. I run the neuromuscular program here. I take care of all the kids with different types of neuromuscular problems, and predominantly Duchenne muscular dystrophy and other types of muscular dystrophies. So we run a robust multidisciplinary program to take care of these boys and also young men and female carriers with dystrophinopathy as well. And we have a robust clinical trial and research program. We have been involved in multiple gene therapy trials and other therapeutic trials here in the Duchenne landscape. And I'm happy to be in this time. We have a lot of challenges, but those are good problems to have in this time of evolving landscape like Pat was alluding to. One of my passion is to help the families and kids that have challenges in accessing care, the best care that they deserve, like everyone else out there. And thank you again for inviting me for this.
Yeah, thank you, Dr. Panda, for that introduction, and thank you for, again, all the work that you're doing, in the community, within the field, with patients. So thank you both for joining us. I want to start first with the patient, really thinking about what does life look like for a patient with Duchenne muscular dystrophy. And so, Pat, I'm going to turn to you and ask, can you tell us a little bit about what that journey looks like from diagnosis through, seeing a pediatric neurologist like Dr. Panda?
Sure. Thanks, Cassie, for the question. The diagnosis is a harsh one, right? The natural history is well known and well described. So you want to find out what is the best care, who is the person to deliver that care. I think in looking for that person or the interdisciplinary clinic that you're going to, you first establish trust. Are these people that are going to listen to you, understand what priorities you have, and are you going to trust them to be able to answer the questions you have, which are numerous and frequent and probably everlasting for most of the physicians? But can you explore with them all sorts of things, even though they are not traditional kinds of therapies? Can you talk to them? Then what is the standard of care? At early ages, as you might imagine, the standard of care is typically a steroid intervention, and that's what we use. Then we have to have those discussions about the long-term effects of steroids. What do they do on behaviors? What do they do on growth? What is their impact on bone? And then the question is that heart built to last? Is that little heart, if we can protect function, is that heart going to be stable? And then you learn the genetics and this whole new exon, intron, nonsense mutation. So you have to learn this other language that is puzzling at best. You look at the sort of pictures of it in the little cartoons, but that doesn't really deliver the sort of deep genetic knowledge that someone has to have in order to really help you and guide you through this process. Then you're looking at what plans did we have? Where do we live? Do we live near one of these centers? Do we have a house that's going to be okay for this child? Because they don't typically do stairs very well. You have to worry about them if they fall, that they might fracture. And so you're worried about their environment. So you have to figure out, what does your environment look like? How do you transport these children? What does the school environment look like? What does that community look like? So I always say, when you get a diagnosis, you become an advocate by design, and by default, because you have to find the doctor. You have to inform your community. You have to inform your expanded family members about what this is. And often you'll get, oh, he looks healthy. And it's hard to describe that down deep, this genetics of this little one means he isn't healthy. So I think the burden of this disease, the expense of this disease, the amount of time it takes, and often the attention that you have to give to this child just to look at his life, make sure he's in the best care, think about participation in a clinical trial, think about what options are on the table and what might be options in the future, and how do you weigh them. I think there's an inherent burden for parents and families, for every member of the family, let alone you're exposed to this pedigree of your family. Is there a carrier of this disease? Are you, the mom, a carrier of this disease? And I will say, I am. And it is an emotional toll that, did you do this to your child? So the mental health disease is another burden for the family. So overall, each day is different, but each day you carry this burden of, what should I do? Am I doing the right thing? Am I doing the best things possible? And so I think we have to look at that whole burden of care, the burden of expense of, what do I do to this house? Do we move? Where do we live? What is my employer? What's that insurance going to cover? So there are so many aspects of this that it becomes overwhelming. But at the end of the day, you go to bed and you pray to whoever you pray to. Don't let it go a step further. Just protect my son and his function where it is today.
Thank you, Pat, for beautifully painting that picture of the challenges outside of what we see in the clinics, right, in the medical setting. But to add another color to what you've just described, there are a lot of families, majority of families that we work with are in the underserved regions, where their Duchenne is not their first problem. I need to work today to get money to feed my family. Those are the families that are untapped into. Those are the majority of our families with Duchenne. And I think they also deserve the exact same care like anyone else deserve living in New York or any other easily accessible areas in the metropolitan cities. But anyway, I just leave it out there. That's one question.
No, Panda, I think you're exactly right. And I think we don't understand that because there are families that have to decide whether I go to clinic or whether I serve dinner for my family, whether I go to clinic or whether I lose my job. So I think that in this rural population, we don't even know the questions to ask. I spent some time in Mississippi trying to understand the barriers to diagnosis, and it is true. Some of it is mistrust of medicine. Some of it is I just can't get there. And one father said to me, these fancy new therapies coming online aren't for me. We aren't in a position to access them. Those are barriers we need to break.
Absolutely. We talk about newborn screen, to diagnose them early on as soon as possible, right? But I'm still diagnosing kids with Duchenne at age 8, age 9. We are still seeing that the journey that used to happen in the past, they're going to the GI doctors, thinking of a liver problem, getting liver biopsies. I'm still seeing that today. So that is a reality out there.
Yeah, I think you both paint a really illustrative picture of how overwhelming and challenging the diagnosis can be, and then from there, the uphill battle of the immense amount of learning that patients and families have to do to really get to a place where they can make decisions for their family and for their kids. And so I think I really appreciate the insights both of you have shared on that. I'd like to shift a little bit to talking about the scientific underlying cause of the disease. And so for that, Dr. Panda, I'd like to turn to you, and if you can describe at a high level some of the genetic components involved in the cause of Duchenne muscular dystrophy.
Sure, I think most of us know that, but just going to briefly talk about it. It is an X-linked disease, meaning it affects the boys, right? But there are also carriers, the moms and sisters, there are female carriers. And this is peculiar in that case that the carriers can also be manifesting, meaning they can also have symptoms of muscle weakness or pain. The gene, the DMD gene, it's one of the largest gene in our human body. It's extremely challenging in the drug development phase, especially for the dystrophin restoration therapies, because of that, how complex and how large the gene is. And it encodes for the protein, which is the dystrophin protein, which is which helps with your muscle contraction, preventing the injury that's happening to the muscle when they contract, and also structural integrity of the muscle membranes. In Duchenne, what happens is when we have these genetic mutations affecting this gene, the DMD gene or dystrophin gene, you won't make any dystrophin protein, or may make some that is not functioning, a normally functioning protein. So that causes muscle damage and causes a muscular dystrophy. But I think what we know now is, even though sometimes we see clinically, meaning when we see symptoms of kids not performing, like not walking normally, or they have problems climbing stairs or running, it may appear around age 3 or 4, or maybe 5 years old, but the muscle damage process happens maybe in utero, even at the time of birth. We know that the CK, the creatine kinase, which is one of the muscle enzyme, a marker of muscle injury that we check for in these boys, which is always high. It's always high from the get-go. So that muscle damage is happening very early on, and I think that's where the newborn screen come into play. How we can intervene maybe early on to further prevent that cascade of muscle damage.
Yeah, thank you for describing that. And Pat, I want to ask you, what does that look like in day-to-day life with a son who has Duchenne muscular dystrophy, as you think about the lack of dystrophin protein and muscle wasting that occurs in various muscles?
Yeah, thanks, Cassie. So we know as parents and family members that dystrophin is absent. And we know because dystrophin is absent, the muscle cell can't survive. So early on in the disease, when this child is growing, there's enough turnover of muscle, and in fact, sort of it tilts toward turnover in terms of how we see these children. So there are times in that early years where you can close your eyes as a parent and say, maybe they're wrong, maybe it's not Duchenne muscular dystrophy. And then at the age of 7 or 8, there's this plateau that they reach where the growth that curve of growth and exaggerated growth or accelerated growth slows down. So then you begin to see, even though in your back of your mind, you know, he doesn't like stairs, he doesn't want to ride a bike, you just see the refinement of small and large motor muscle. But then when he plateaus, then you see even more evidence of the disease process. Or when he goes with friends who come over to your house and they're running around your house and up and down the stairs, or even jumping on the furniture, which we don't accept, except, you know, your sons aren't going to do that, right? Because they can't. They are not risk takers typically. So for parents, because dystrophin is missing, the fundamental desire is get it back however you can get it back a little bit, even a little bit that parents would say is reasonably likely to create benefit, right? So even if you get me 1%, I'll take it and run. But obviously then we see a more milder form or a different form of muscular dystrophy on the other end of the spectrum called Becker. And those folks do have dystrophin. So you want to go there. So fundamental for the parents is get dystrophin back, however you can, short, long, whatever, in a certain percentage. And so I think parents are looking toward that as a priority, and all other therapeutic development would be supportive of that dystrophin expression. So I think that's what parents look at. And when they see that plateau and they know the natural history is here, it confirms the disease course, it confirms the progression. And so we are looking at that and watching that CK as a marker to say, things are still going downstream when we need them to slow down.
Yeah, so Dr. Panda, from your clinical perspective, whenever you have a DMD boy, you know, that is in your office and you're talking through treatment options of what's available, you know, standard of care treatment today, what does that look like? What kinds of treatments are available for these patients?
Sure. You know, like we talked before, the treatment landscape for Duchenne is evolving so rapidly, right? So we have our foundation is still our corticosteroids. We have years and years of experience now using steroids that we know it slows down the progression of the disease, right? We always talk about skeletal muscle, but also there's your heart is also a muscle, so heart is involved, your breathing muscle is involved, and we know the corticosteroids can delay the onset of what we call cardiomyopathy in these patients, can overall slows down the progression of the disease. So that's the foundation that we talk about. Now we have three options, right? You have prednisone, you have deflazacort and Emflaza, and then you have the most recent one, which is vamorolone or Agamree. So I think that's the first discussion: steroids, not steroids, which dose to use, and which steroid to use, and what dosing. I think that's just the first discussion. And then depending on the boys after that, then we talk about if they would be eligible for one of the available exon skipping therapies. We'll talk about that. We also talk about Duicet, which is recently approved. I think the challenge is also making decisions which one to go first, how to stack therapies. We have options now. So I think that's how the discussion happens. Now, with the new diagnosis, it's already discussing about the diagnosis, and just the disease itself is overwhelming. It takes sometimes 2 to 3 consecutive visits with this to actually get into these treatment options. Then we cannot ignore the clinical trials that's also ongoing. That should be part of the discussion also. So I think it takes a few visits to discuss these options. But I think today it's still steroids. I don't think we are there yet to say we don't need steroids anymore. So that's the foundation we talk about, and then how we can add on either exon skipping or givinostat or other clinical trials.
Yeah, I think this is a good opportunity as we're thinking about the clinical trial landscape to take a few minutes to introduce the program that we're developing here at Precision that we call PBGENE-DMD. And our goal really is to be in our clinical study early next year in 2026. So the team at Precision is working literally day and night right now, finishing up all of the requirements to submit our CTA or IND later this year with a plan to start that trial in early 26. So I just want to take a couple minutes to introduce this program. So as you can see on the slide here, PBGENE-DMD is a gene editing therapy that we've designed here at Precision with the intention to provide durable functional improvements for up to 60% of patients with DMD, those who have mutations in this particular region of the dystrophin gene between exons 45 and 55. We know up to about 60% of patients have disease-causing mutations in this region. And so what we've done at Precision is we've utilized our proprietary ARCUS technology, our gene editing technology, to develop two nucleases. Both of those nucleases can be delivered in a single AAV. And once that AAV is internalized into muscle cells, those nucleases recognize binding sites surrounding exons 45 to 55. And whenever they create a double-strand break at their target sequences, it results in excision of this region of the dystrophin gene and resulting in an in-frame now dystrophin gene, bringing together exons 44 and 56. And this results in a functional protein that can be produced, a near full-length protein. And we know that this protein is functional in humans because there are Becker patients that have this genotype, this del45-55 genotype, who produce the same protein, same dystrophin protein that our PBGENE-DMD editing approach results in. And those patients generally have a more mild form of muscular dystrophy. Some of them are even asymptomatic. And so we really intended to develop this program to be, a single AAV construct that can deliver long-term functional improvements. We have completed a long-term durability mouse study where we administered PBGENE-DMD into a diseased mouse model, a DMD diseased mouse model. And so what you're looking at here is functional data from these mice. That this experiment was carried out to 9 months, so a long in-life study in mice. And we measured force output at 3, 6, and 9 months. And you can see here in the light gray bars, these are the DMD mice. And so this is their baseline force output at each of these time points, all normalized to 100%. And the darker gray bars next to them are our control animals. So these are healthy animals that express full-length dystrophin protein. You can see they, at all of these time points, demonstrate a substantially improved ability to exert force in the gastroc in these mice. And then the last set of columns are from our PBGENE-DMD treated mice at 2 different dose levels, 1E14 and 3E13 VGs per kg. And at both of these dose levels and at all of these time points, we see a substantial improvement in the ability of these mice to exert force output near almost healthy levels in these mice. And so this was a really exciting result for us at Precision. This was really the first evidence that PBGENE-DMD can demonstrate long-term functional improvement in this preclinical mouse model. And while we won't go through the data today, this functional data is supported by molecular data in the tissue demonstrating a significant increase and durability of dystrophin protein expression over the course of this study, as well as % positive dystrophin cells. And what we were really excited about is that we were able to demonstrate the editing of satellite cells within the tissue. And we'll talk a little bit more about the importance of satellite cells as it pertains to a gene editing approach in a minute. But these are the cells that give rise to new myocytes that help replenish the myofiber, as we talked about, as particularly important in Duchenne patients that have this muscle fiber wasting over time. Those satellite cells are what help replenish the tissue. And so being able to edit in the satellite cells, we think, is really essential for thinking about long-term dystrophin protein expression from a gene editing approach. And so at Precision, we're really excited that this approach could be broadly applicable, could provide long-term durable functional improvement for patients. And we're excited about the preclinical data we've generated thus far and excited to bring this to clinic, as I mentioned, in early next year. And so I think first I'd like to ask you, Dr. Panda, you've interacted with us at scientific conferences. You've seen this data at MDA and World Muscle Society. What's your impression of the PBGENE-DMD program, and how do you see it fitting into the clinical trial landscape?
That's the preclinical data is amazing, Casey. But before I answer your question, can I ask you a question?
Of course, please.
Can you educate us how the ARCUS technology, what's the uniqueness about it, what's special about it compared to some of the other gene editing technologies?
Yeah, of course. Great question. So at Precision, we developed the ARCUS technology, and ARCUS nucleases are a completely different type of gene editing platform compared to CRISPR-based systems, which many people are familiar with, even down to the origin. So ARCUS nucleases come from a green algae versus a bacteria like CRISPR-based systems. What's really, I think, advantageous about using ARCUS nucleases for gene editing, particularly in this program, is that ARCUS nucleases are very small. So they are only about 1 kb of sequence. You can fit both of those nucleases into a single AAV. We know AAV dosing is really important to maintain safety. Keeping that dose as low as you can is important for an overall safety profile. So being able to put all of the components necessary to complete this 45 to 55 excision in one AAV, we felt like was really essential to be able to bring this product to patients. The last thing I'll mention about the ARCUS technology that we think is important for this program is that the nucleases create a 4 base pair 3' overhang. And the nucleases that we designed for this program actually have compatible overhangs. And so after you create those cuts, those two pieces of DNA can fit back together like a puzzle piece. And that makes that religation more predictable and more efficacious, more efficient in terms of creating that therapeutically relevant, therapeutically necessary edit. And so we really like the ARCUS platform for DMD gene editing for this type of an excision approach. I really appreciate the question.
Thank you. I think it's always helpful for clinicians and also for the families to put in a context of like what's the difference, what's unique about each approaches. I also want to add, I think we talked about the efficacy, how it could help with the efficacy part of it, but can you also shed some light on any information that you have regarding the safety? Because at safety comes the priority. When we talk about gene editing, we all scared about the safety aspects of it. If you can shed.
Yes, of course, absolutely. We agree. Safety is absolutely paramount, particularly when you're talking about permanently editing the genome. So ARCUS nucleases, as I mentioned, are derived from a green algae versus other editors are derived from human pathogens like CRISPR-based systems. And what's been demonstrated in the literature is that there are humans in the population who have pre-existing exposure to Cas proteins and have developed immunity against Cas9. And so that's been a real challenge, I think, for thinking about something like an AAV delivery where you're going to expect longer-term nuclease expression. If there's pre-existing immunity, that could be a safety concern. So I think that's reassuring from the ARCUS perspective that the nucleases here are not derived from a human pathogen. They're derived from a green algae, as I mentioned. And we have looked within human donor PBMCs to see if there's any existing pre-existing immunity within the human population. And as you'd expect, since it's not a human pathogen, there is not. And so I think that's a major differentiator for our technology. And then in terms of the safety profile for PBGENE-DMD, we've just recently actually completed our non-clinical studies to support this program. And at Precision, I'm really proud that we do a comprehensive analysis of the safety of our nucleases for specificity, as well as the overall tolerability in terms of our GLP studies, including in non-human primates. And so I think we're feeling really confident with a lot of the safety data. We haven't publicly disclosed much of that data yet. We hope to do that in the future. But so far, I think it's all signs supporting initiating the clinical study.
Thank you, Kathy. I think going back to your question, I think from a clinician standpoint, I think what we know right now is the landscape is evolving with a lot of learnings from the approved therapies as well as the clinical trials that we are doing, and I think which is great. Right, I still don't think the therapies that we have is a cure, right? So we are always looking for what's next, what's better. So from that perspective, I see your program as one. I think it's based on the target the exons that you're targeting. It can be applicable for most of the patients or boys with Duchenne. not all, but I think majority of them, right? And I think I hear that there might be, even though we're using AAV9, our dose might be lower than what is being currently used in the microdystrophin side. I think that's a positive thing I'm noticing here as well. And I see this also as a multiple, even though it's a gene editing technology, but I would see that the endpoint is like more of a multiple exon skipper, which is permanent. And it is good to see the cumulative effect on your biomarker data that you just presented, as well as the functional data. And then another unique also is the satellite cells, which we already highlighted on how important it is if the gene editing is happening within the satellite cell. And if you can show that consistently, I think that's going to be a huge game changer as well.
Yeah, thank you for sharing your perspective. Pat, I'd like to ask you, from a family perspective, how do you think about the clinical trial landscape, and where do you see PBGENE-DMD fitting in?
So, Cassie, we've certainly had a long history of trials, and as I mentioned, 8 approvals in the U.S., and those approvals have really informed us, and informed us in terms of being able to modify disease progression. And so they have done exactly that. And we're learning in trials that it's a cumulative effect, that muscle doesn't just turn around the next day from an intervention, but it's a cumulative effect. What I think as a parent you're looking for is, we are thankful and grateful for all of the work that's been done, all of the young men who've participated, and some women, and we've built on their shoulders and built on the shoulders of industry who's come before us. I think what we're looking for now, and what we feel like the landscape has so evolved, that PB gene therapy, looking at editing the hotspot, if you will, from 45 to 55 for 60% of the people, would be one of those permanent, not just disease modifying, but really transformative therapies, where these children, or these young men, depending on their age and stage of disease that were treated, would have the confidence that not only they are expressing a safe and effective and stable protein, that is reasonably likely to really stop that disease progression. And that would be such a breakthrough and such an amazing progression in this landscape of evolving therapies, that would really give us that light in this tunnel that said, we're now just not modifying, we are really stopping disease progression. And as I mentioned before, they're thankful to all the blood, sweat, tears, and time that these families have put into it and continue to put into it, looking for a therapy that is more than modifying, that is transformative, and that they can go to bed every night and say to themselves, it stopped, which would be, similar in cancer when they say NED, no evidence of disease. I don't think it would be no evidence of disease depending on the age and stage, but boy, going to bed and saying, this is life-saving, life-changing, would be a real gift.
Yeah, it's really inspiring to hear you talk about the progress in the field. And, at Precision, we have paid very close attention to the therapeutic development space, trying to take every learning possible from those who've come before us to really set our trial up for success. And we're really appreciative of all of the families and all of the science that's gone into really the decades worth of work that's gotten us today to where we are, that we're excited to bring this really first-in-class gene editing approach into the Duchenne field, and just really grateful for all of those who've come before us and that we've had the ability to learn from. So, Dr. Panda, I want to ask you, as you think about the clinical development progression from here, what do you think is most important for Precision to be focused on at this moment while we're getting ready to start our clinical study next year?
Yeah, I think that's. I have a lot of suggestions for that, which. I think, yes, it's important to have a rigorously designed clinical trial to show the effectiveness of the drug itself, or whether it is safety or the effectiveness, right? But we also have to keep in mind, we are asking a lot from the families that we're working with. How can we keep it to minimum possible with the exact same rigorousness, control, but minimum that we could ask from these families? How can we support the families throughout the journey of participating in the clinical trial? I think that's most important to me. You can list number of assessments. You have to get the North Star or this and that, and you have to do it every 3 months. Is it really important? And then another thing that to minimize the burden on the families and whatever we can make it easy for them. I think that's the most important thing that I would say. I think number two, how can we strategically plan to safely dose this? It's not just the dosing, but monitoring after the dosing also, right? How can we do that safely? How can we make sure that these families have access to the experts and whatever help they need, whatever expert they need? We have to ensure that when you do these trials, they have access to all of that. So we ensure the safety is the utmost priority.
Yeah.
Another plugin is also how can we get these inaccessible families get into participate in the clinical trials? We are not doing a good job of that so far. And I think it needs a lot of work and a lot of strategical thinking and planning from a sponsor side, as well as we are, I'm sure Pat and the sites will be willing to work with you on that. But you're fighting against how fast we need to enroll them fast so we can get it to the market faster, get it to be available to everyone faster. But I think it needs a little bit more thinking. Those are my thoughts. Yeah.
All good advice. I appreciate you sharing your thoughts. And Pat, I'd like to ask you, how do you see the role of advocacy groups in partnering with industry sponsors like Precision to really help set up a successful clinical study?
So I think the advocacy groups bring a wealth of experience and knowledge. And I think many of the people in advocacy groups such as ours are parents. So they've been involved in a clinical trial. They know what we're talking about, burden, what that means from the being screened and thinking about the right trial, whatever that right trial is, as guided by their clinician. Then thinking about the informed consent. I will say to you, I often say it's neither informed nor consent. It's the ticket that you wish for that provides benefit for your child. So we all sign a document, and I know it's a very legal document, but I'm not sure we care deeply about what it means, even though I know. So I'd have an executive summary. Here are the 3 points you need to pay attention to, because 20 pages is way too long. I think being very careful about what are the implications, what are the need to have versus nice to have kind of outcome measures. Putting a child through 10 outcome measures in a single day, I can't imagine that really represents real life. So how can we think about looking at real life? What is the real world data these parents experience at home? What are they seeing? Because that often what the parents see, what is meaningful to them, may be different than is meaningful in terms of an SAA, the North Star, right? So what are you seeing at home? I think also recognizing that this child is different in terms of his schooling or his family. So he's a child with a diagnosis, and participating in a trial, taking away more time from school, more time from his family, makes him more different. So I think rewarding that and understanding and being able to have some psychologist or one of the folks that are in these interdisciplinary clinics, I think they're called life science people, talk to them and recognize this is hard and you're away from school and you're missing your friends and probably the best TV show you've ever seen. So I think that's it. Also, I think you have to involve this as a family disease. If there are other children at home, having an educational document so that they can read it and understand what my brother is going through, and also for the grandparents, because sometimes they're called in to do the sitting or the running around without real knowledge of what's going on. So I think these are small things we can do to reduce burden. I think that along the way, at least recognizing that they are putting in all this time. And I don't know that regulatory will pass this, but placebo-controlled trials, when you're looking for dystrophin and biopsying them, make zero sense to this community. If you're trying to get dystrophin expressed and it's not there and you've got none on biopsy, I see no reason to do a placebo. And we are happy to work with you, with our regulatory authorities, to say that doesn't make any sense. In addition to the fact that there are side effects giving a virus, so that AAV carries nausea and vomiting with many of the patients. So we're very clear to know who's on placebo and who's not. So I think this is a team sport, and I think industry needs to recognize advocacy plays a role and patients play a role, and together we can develop what's need to have in these trials, what isn't necessary in the trials, and when will you tell me how my son did in this trial? Because all of these parents want to know want to know the data. How did everyone do? But how did my son do? So I think being respectful of this community and its participants and advocacy is really going to ensure trust of your approach, trust of the medical profession, and a willingness and really thrilled about the clinical trial you intend to deliver to us.
Thank you so much, Pat. We're so lucky at Precision to have, partners like both you and Dr. Panda helping guide our clinical development plan. And we so appreciate all of the input that you have shared with us as we are planning for our clinical study next year, with the goal of really, hoping to be that transformative therapy for these patients. That would just be, I think, a dream come true for all of us. It's just an amazing outcome. And so I want to take a moment to thank both of you for participating. It's been a really inspiring and enlightening conversation today. I want to end by just asking if either of you have any final thoughts to share with our audience as you think about what lays ahead over the next months and years for the Duchenne community.
Pat, do you want to go first?
When I started PPMD 30 years ago, this was the dream, right? This was the hope. This was the light in the tunnel. And I think getting to a transformative therapy is exactly the light that this community needs and deserves. We've waited a long time. We've built on the shoulders of giants in terms of people who've come before us. And now you've set the stage, and I wish you Godspeed.
Thank you, Pat. Panda, anything to add to that?
Nothing to follow Pat's wisdom, but just one thing to add from a clinical trial planning standpoint, which Pat already touched upon, is the data sharing. I think that and not making our boys repeat things unnecessarily. How can we integrate the clinical trial testing, like if you're planning on cardiac MRA or skeletal muscle MRA or even assessments that we do, make that available for their standard of care, their clinical care, so they don't have to repeat these all over again next day or a week later.
Yeah, that makes sense.
Yeah, I'm excited to be part of this program.
Yeah, we're so grateful again to both of you for all of the support, all of the insights that you've shared over the years with us, and as we gear up for this really huge moment for Precision. And I can tell you with confidence there is a team of dedicated folks here in Durham, North Carolina, who are so excited to bring this program over the line to patients, and just really, again, appreciate all of the support from the community that we've had, and excited to bring this to patients with our CTA or IND later this year and clinical studies starting up next year. So with that, I think we'll close the conversation. Thank you again to both of you for participating and to all of our audience members for tuning in.
Thank you.